This video is a review of the concepts of Part 3 of the course MCDB 101B, Winter Quarter 2016. Starting with the second half of Lecture 18, the human genome consists of approximately 20,000 protein-coding genes and 5,000 or more non-coding RNA genes for tRNAs, ribosomal RNAs, small nuclear RNAs, and microRNAs. Only a small fraction of the human genome are exons that encode for protein. Most of the genome is quote-unquote junk DNA whose function is unclear. This junk DNA includes introns, intergenic regions, and non-functional transposable elements. Lecture 21 covered two important technologies that have made modern molecular genetics possible. They are PCR and DNA sequencing. PCR, or polymerase chain reaction, amplifies a small amount of DNA to give us a large amount of DNA using in vitro DNA synthesis to double the amount of DNA every round. For the final, you need to know how to design PCR primers to amplify a given region of DNA. DNA sequencing specifies our genotype by providing our complete DNA sequence. We learned about the older Sanger dideoxy sequencing method as well as the modern sequencing by synthesis. Be familiar with both technologies at a conceptual level that allows you to answer the quiz iClicker and practice midterm questions. The human genome is sequenced by a strategy called shotgun sequencing, in which random short regions are sequenced and then the final sequence is assembled using sequence overlaps. In Lecture 22, we learned that the vast majority of human traits are non-Mendelian. They are complex traits. Complex traits are multifactorial, meaning that multiple genetic and environmental factors contribute to the trait. Contrast a complex trait to a classical Mendelian trait in which a single gene completely determines the trait. Complex traits are quantitative and continuous, and so the phenotypes span a range of values, and these values typically form a normal or bell curve distribution. A normal distribution is described by its center or mean and its width, which is represented by its variance, a number that you're expected to be able to calculate given the formula. The fundamental law of genetics is that phenotype equals genotype plus environment. Can we formalize this law into a more quantitative statement? The total phenotypic spread or variance in a trait in a population is equal to the phenotypic variance arising from different genotypes, termed the genotypic variance, or V sub G, plus the phenotypic variance arising from different environments, termed the environmental variance, or V sub E, which leads to the equation V sub P equals V sub G plus V sub E, or that the total phenotypic variance equals the genotypic variance plus the environmental variance. Heritability is the fraction of the trait that is due to genetics or genotype and is equal to V sub G over V sub P, or in other words, the genotypic variance over the total phenotypic variance. Heritability is a number between 0 and 1. The heritability of a classical Mendelian trait is 1. In other words, the trait is completely determined by genotype. We can estimate heritability from twin studies. There are two basic types of twin studies. First, you can study identical or monozygotic twins who are separated at birth. There, heritability equals the phenotype correlation coefficient for the monozygotic twins separated at birth. Intuitively, the greater the heritability, the greater the correlation in the trait for the identical twins separated at birth. Second is the classical twin study, in which you compare the phenotype correlation coefficients of identical twins reared together in the same household with fraternal or dizygotic twins reared together. Heritability equals 2 times the difference between the monozygotic twin phenotype correlation coefficient and the dizygotic twin correlation coefficient. Intuitively, the greater the heritability of the trait, the greater the difference between the two correlation coefficients. In Lecture 23, we learned that a SNP is a single nucleotide difference between different people in the population. I may have a T-base pair at a certain position in my genome sequence, and you may have an A nucleotide. A SNP is polymorphic if different people tend to have different alleles of the SNP. Most SNPs are biallelic. SNPs are very useful as genetic markers because they have a specific location in the genome as well as different alleles. Complex traits are influenced by multiple genetic factors. What are the genes that contribute to a trait? We have used two different approaches to identify these genes. Number one is linkage analysis, and number two are association studies. First, linkage analysis. The idea of linkage analysis is to determine if the SNP marker is close to the disease gene by calculating the recombination frequency between the two using data from a pedigree. Remember, we don't do experimental crosses with humans, and so we need a pedigree to examine recombination events. First, assume that the disease trait is conferred by a dominant allele in the pedigree. Then, examine the progeny of a double heterozygote who is heterozygous for the dominant disease allele as well as heterozygous for a nearby SNP marker. The progeny can then be scored as parental types or recombinant types, which can be used to calculate the recombination frequency. The statistical significance is determined, as we've done before, by calculating the chi-square value for the null hypothesis that the marker and disease allele are unlinked, and then trying to reject that null hypothesis with a p-value less than 0.05. Basically, we're applying the same techniques as we used previously for 2-point and 3-point crosses. The association study can be thought of as a generalization of linkage analysis to the whole population instead of pedigrees. Two alleles that are close together on the same chromosome are in linkage disequilibrium. They tend to occur together or associate together in the population. In other words, a particular haplotype may be enriched in the population. A candidate gene study, one looks at the association between a given SNP marker, typically close or in a particular gene, and a disease trait. One performs a case control study by genotyping the case or affected group, as well as genotyping the control or unaffected group for the two SNP alleles. If there is an association, then one of the two SNP alleles should be enriched in the affected population. The data are laid out in a 2x2 contingency table, and the association can be quantitated by calculating the odds ratio, which you should know how to do. The statistical significance of the association can be determined by our chi-square test calculated by comparing the observed data in the table to the expected data. As demonstrated in class, you should know how to determine the expected 2x2 case control table. The null hypothesis is that there is no association between either allele and the disease trait. We calculate the p-value to determine whether we can reject the null hypothesis and then conclude at the 95% confidence level that there is an association between risk allele and disease if p is less than 0.05. We can extend this analysis by looking for association between the disease trait and markers in every gene across the genome by performing a series of association tests for each marker. This is called a genome-wide association study, or GWAS, and may involve as many as 500,000 SNP markers. To test for significance, it is important to correct for multiple testing and calculate a new p-value threshold, which is 0.05 divided by the number of tests. This is the Bonferroni correction. Turning to lecture 25, although at the sequence level we share greater than 99.9% of our DNA at the sequence level, the amount of sharing at the gene allele level may be less. For polymorphic genes possessing many alleles, most people will tend to have different alleles. For a polymorphic gene, you share approximately 50% of your alleles with your parents and with your siblings. You share approximately 25% of your alleles with your grandparents and 25% of your alleles with your aunts and uncles. Extending the family tree to all humans is the goal of ancestry studies. The key to ancestry studies is the patrilineal inheritance of the Y chromosome and the matrilineal inheritance of mitochondrial DNA. From a hypothetical founder male, which is termed Y chromosomal Adam, the Y chromosome has been passed down through the generations from father to son to all males living in the present. Likewise, mitochondrial DNA has been passed down from a hypothetical founder female, termed mitochondrial Eve, from mother to daughter to all females in the present. The only changes in the Y chromosome or mitochondrial DNA result from mutations. Single nucleotide mutations, or SNPs, can define different haplogroups based on which allele an individual has at the characteristic SNP. These SNPs can be used to construct a cladogram in which each node represents a common ancestor as well as the characteristic SNP. And each branch from the node represents one of the two alleles of the SNP that was passed on to different haplogroups. Make sure you understand how to interpret and reconstruct the cladograms and haplogroup genotypes. Finally, turning to lecture 26, researchers can manipulate the genome and genotype of an organism, thereby creating transgenic and genetically modified organisms. We can add a transgene, we can knock out or delete genes, we can edit genes to correct mutations using the CRISPR system. It is possible to alter the genotypes of humans using gene therapy to try to correct genetic disorders. Viral vectors are used to introduce a transgene to human cells, and as an example, I describe introducing a wild-type version of the beta-globin gene into red blood cells of individuals suffering from beta thalassemia, in which both copies of the beta-globin gene have been mutated. There is a big distinction between somatic cell gene therapy and germline gene therapy, or gene editing, in which the genotype changes can be passed on to the next generation. In lecture 28, we covered in lecture, 28, is that right? Yeah, lecture 28, we covered cancer. Cancer arises from the altered genotype in cancer cells. Multiple hits to the genome are produced from multiple somatic mutations, as well as some inherited mutations. These mutations, which include chromosomal rearrangements, affect two major classes of genes. First, gain-of-function mutations, such as constitutively active mutations or mutations that overexpress the protein. These gain-of-function mutations turn on oncogenes that promote cancer. Many oncogenes are involved in growth factor signaling that leads to cell proliferation, and the activating mutations allow cell proliferation in the absence of growth factors. Second, loss-of-function mutations, such as null mutations, inactivate tumor suppressor genes, which normally protect the cell from cancer. One classic example of a tumor suppressor gene is the retinoblastoma gene, which when inactivated can lead to retinoblastoma. Finally, personalized medicine encapsulates the idea of tailoring treatments and diagnostics to the individual patient instead of a mythical average patient. Genetic testing involves determining your genotype for genes that contribute to a disease. For example, BRCA1 is a tumor suppressor gene, and loss-of-function mutations in BRCA1 lead to an increased risk of breast cancer. Early onset breast and ovarian cancer ran in Angelina Jolie's family, and she used a genetic test to show that she had a loss-of-function mutation in BRCA1 that conferred a higher risk of breast cancer. As a result, she had a preventive double mastectomy performed. And this concludes our summary of the concepts in Part 3 of MCDB 101B.